Good morning, everyone. We're going to go ahead and get started in the interest of time. For everybody on the live stream or who will be watching this later on, my name is Seth Hamby. I'm the Living Collections Director here at the Garden, and I have had the distinct privilege of supervising our summer volunteers. We have Anastasia Barron, Julian Moore, and Megan Boneck. Anastasia has been working with the Begonia Collection doing photography. And then Megan and Julian have been working on our Woody vouchering program, and you'll hear all about those things. I'd like to give a shout out to the people who have helped mentor the students over the summer. So we have Benjamin Dorrington, who's our plant records coordinator. Sorry, Benjamin. And then Austin Brennick, who is our assistant plant records coordinator, and they have been working with Julian and Megan over the summer to work on the Woody vouchering program. And then we have Glenn Dickerson, who's the curator of the Begonia collection, who's been working closely with Anastasia and her photography project. We also have a volunteer mentor who's not able to be here today. His name is Victor Mosqueda. And he has been also very influential in the whole process, helping with the Begonia photography internship. So I want to turn it over to Anastasia to get started. So please welcome Anastasia.
Hi everyone, as you said, my name is Anastasia Barron. This summer I was an intern here at the garden working to scientifically photograph the Begonia collection. Before that, I want to tell you more about myself. This fall, I will be a senior at the University of North Texas, pursuing a major in ecology for environmental science, and I have a completed minor in photography. However, I am debating on going back to complete a bachelor's in photography because I wish to incorporate it more into my future career. So that's on the fence. I love plants and photography, so I was very excited to hear about this internship because it's kind of the perfect match for me of my two main interests. And it was so fun working with Glenn and everyone in the Living Collections team. And that's little me with my grandma. She's the reason why I love plants and photography, so I thought I'd put a picture of us together in here. And I'd also like to give a big acknowledgement and thank you to ABS and ABS Southwest, as well as IMLS, for providing the support and grants that we needed to pursue this project. Without y'all, this would not have been possible. So I'd like to give a big thanks to y'all. And I'd also like to give a shout out to Victor Mosqueda, who was mentioned earlier, who cannot be here, Glenn Dickerson, the Living Collections team, and of course the Fort Worth Botanic Garden for supporting me and for providing this opportunity to meet to begin with. So, first off, what is the Begonia Species Bank? So here at the garden, they have a species bank of begonias that has about 609 species along with 665 cultivar species. This collection began in the 1970s with Mae Blanton and her friends and was largely developed by Debbie Garrett in 2007 up until her retirement, at which time Glenn took over. And so over the summer, we worked to develop a project of scientifically photographing begonias using a technique called focus stacking. This is a digital imaging technique that allows us to create really high quality images where the entire plant is in focus even when zoomed in. And over the summer, out of the collection, I managed to photograph 34 of the 609 species. That's about 5.5% of the total collection. So it's a very slow going process, unfortunately. Okay. So what's the purpose of us taking these photos? Well, the main goal is to create an online database of begonias that is accessible to the public, whether that be for research, educational, or just curiosity purposes. We want it to be super user-friendly and to provide consistent scientific photography to those who need it for whatever reason. And this is a photo that I took of Begonia albococcinea hook. And so there are some current databases out there that provide information about begonias. On the next few slides, I have ranked them from most informative to least informative, in my opinion, as I searched and navigated around the websites. So the first one is iBegonia. I classified this as the most informative because it included pictures of all the species that I searched, even if they were sometimes low quality. It gave the viewer a general idea of what the plant looked like. And they also provided descriptions of what the blooms and the texture of the plant was, as well as distribution area. So I found this one to be super informative and easy to navigate. The next on the list is the Royal Botanic Garden Edinburgh. This one was also pretty user-friendly. However, it did not include that many pictures for the species that I searched on here, and the photos that it did include were mainly of mounted cuttings that they had taken, so they were not accurate representations of what the plant looked like in terms of color and leaf texture. But it was great for distribution area information, as you can see. It provides coordinates and country names, etc. There we go. So the next one is the World Flora of Plants Online. This was one of the lesser ones. It was not easy to navigate. It provided absolutely no photos of any of the plants that I searched. It was also missing a lot of species. Whenever I was going through the list of available species that it had, I could not find any of the ones that I had photographed over this summer.
Everyone has the same nose too.
Soon. Royal Botanic Gardens Q. This one was ranked least informative because it had no pictures, no descriptions of the plants, was kind of confusing to navigate and search through, and only really had information about the classification of the plants and of distribution area. So what will be different about the Gardens database? Well, the main goal is to make it more user-friendly, so it's easier to search on the website and to make it easier for people to find what they're looking for. And to, of course, provide images that are of higher quality. So for those who can't come in person to see the begonias at the garden, they can at least zoom in really close on the picture to hopefully see the details that they need to see. And in the future, we hope, In the future, we hope to incorporate more details about the begonia, such as close-ups of the flowers and the blooms and the leaf venation, color, and texture, which you can kind of see a glimpse of in the posters over there to your right. So I mentioned earlier that we're using a technique called focus stacking, but what is focus stacking? Focus stacking is a digital image processing technique where we take multiple photos at different foci distances and we combine them into one resulting image. And this one resulting image is very high quality. For the portraits that we took of these bigger plants, we could take anywhere between 2 photos up to 30 photos. But for the microphotography, those can take upwards of 50 to 200 photos. It just depends on how close we are to the subject. But we use a program called Helicon Focus to combine these images into one. And the more images that we took of one plant, the higher quality the final image would be. So this is a screenshot of the user interface of Helicon Focus. This is what I would see when I was stacking images. On the left is an individual image that was used to create the final one. And on the right is the final stacked image. So if we were to zoom in on that image, theoretically, we would be able to see everything in focus. But on the left, only the front part of the plant would be in focus. And so we are dealing with a lot of files for this project since we're taking so many pictures of each plant. As I mentioned before, we're taking anywhere from like 30 photos of a plant. And so I had to develop a file organization system for whoever needs to access it in the future. And that starts with the classifications of the plants. There are 8 classifications, cane-like, Rex cultorum, rhizomatous, semperflorens, shrub-like, thick-stemmed, trailing, and tuberous. And I also included an extra folder for species that were only in terrariums. This summer, I only got to work with rhizomatous species, so that's the folder that we'll be going into. And when we click into it, we get an alphabetically ordered list of folders, as well as an uploads folder. The uploads folder we used whenever we were transferring images from our camera to the computer. This just made it easier for us to sort into their species folder later on than individually uploading them into individual folders. So if we click on the A folder, we are given all of the species starting with A. And then if we, go ahead. And then all of those species folders contain the same 8 folders. There is one for each side of the plant. So portrait 1, 2, 3, and 4, the top image that we take of the plant, the angle image we take of the plant, the focus stacked images, and then the final edited images. Those same 8 folders can be found in every species folder. And for every folder except for the stacked images and the final edit images, they are further separated into black and white because we are taking images on both black and white backgrounds. So we want to just further separate that for easiness. And then for the focus stacked and photoshopped images, we saved unique names solely for those files. We figured it would be too time consuming to go in and edit the names of every single image that we take. So we saved unique names for only those. For this system, the first part of the name consisted of what the photo was of. So is it portrait 1, portrait 2? Is it of the top of the plant? Is it of the angle of the plant? And then it was followed by a B for black background or a W for white background, followed by FS for focus stack to indicate that it was focus stacked, followed by the species name. And then for the photoshopped images, we just changed the FS in the name to PS for Photoshop. So that's the only difference between those file names. So I used a Excel sheet provided by the Living Collections to keep track of my process as I was taking photos, because it is very easy to get lost in what photos you have taken and what photos you have not. It's pretty simple. It's just a tally system. I created columns for the white portrait, black portrait, white top and black top, as well as the steps of stacking and editing. So for example, if we look at Begonia acetosa vale, at the time this screenshot was taken, all of its portraits had been taken, but it still needed to be stacked and edited. So that can be used for future people to refer to check my process. And then this is some of the equipment that we used when we were taking the photos. We obviously needed a tripod because any movement would have messed with the final image stacking. So we used one from ProMaster. The camera that we used was a Nikon D850 with, I believe, a 24 to 70 millimeter lens. And we used a Faisal tripod attachment for the overhead images that allowed us to suspend the camera directly over the plants. And then for our setup, we used Amaran light domes. These provided consistent soft lighting to the plant to create an even background. And for our background, we used a paper roll. We used a black one and a white one, and that stand in the top left allowed us to hang it, suspend it in the air, and simply roll it out onto the table. And we would use that air duster to remove any dirt or dust that settled on the table because we wanted a clean, even surface. And the room we worked in was very dusty and dirty. So that was essential. And these are some photos of our setup. On the left, you can see a photo of the portrait setup, the general portrait setup with our lights on and the paper roll pulled down. And then on the right is what it would look like whenever we were taking our overhead photos. You can see the tripod with the attachment and our counterweight to prevent the camera from falling forward onto the plant. So the early stages of this project were very slow going, as it is most of the time with photography. We were developing our processes and our techniques and perfecting our focus stacking and learning how to properly focus stack. So due to the trial and error of learning these new methods, it was very slow, and we did have to reshoot some of the plants. That's just something that we'll even encounter in the future because plants are living organisms, they'll move. It's something that we'll have to deal with, unfortunately. So to get into finally the photo taking process, this first starts with preparing plants for getting their photos taken, similar to how a model has to be prepared for like a runway photo shoot with their makeup done and stuff. We have to make the plants look pretty. This involves cutting off any dead blooms or leaves and wiping off leaves if needed. So for example, this is Begonia falcifera. As you can see on the right, it had a lot of what I assume to be pesticide buildup on the leaf that caused it to get like a pale, like dotted texture. And we do not want to capture this on an image because it is an inaccurate representation of what the plant looks like. And it could be misleading to anyone who's trying to use it for research. So I had to go in and wipe all of that off myself before I could take photos of the plant. So the photo taking process, there are 6 photos we take of each plant, both on a white and a black background. The first images are the portraits. So we take it on a white and we take it on a black. This would be classified as portrait 1 because that is the first side we're taking the portrait of is considered the front. So that is portrait 1. And then we can go on to portrait 2. Between each portrait, we are rotating the plant counterclockwise 1 step. And then we rotate again, we get portrait 3. We rotate again, we get portrait 4. And then once we take the portraits, we can take the angle photo. This is where the camera is slightly above the plant and is angled down at a downward angle, about 30 or 45 degrees. It allows for more depth of the plant. And then we can take our aerial photo or the top photo. This is where we use the FISO attachment to suspend the camera directly over the plant. And we had to remain pretty consistent with what we were doing with the angles and such. As you can see, they're exactly the same from black and white backgrounds. That's something that we had to keep in mind when taking these photos. And then, so why are we taking photos on a black and white background? Well, it's because the different colored backgrounds display different details on the plant. So as you can see on the left, the black background, you can see all of those white little hairs on the edges of the plant, but on the white background, you can only see them on the parts that are closer to you. You can't see most of them on the back. So those are details we have to keep in mind that we want to capture since we're trying to accurately represent these plants. So after we take the images, we begin the editing process. This process, we use Helicon Focus, Adobe Lightroom Classic, and Adobe Photoshop. The process started in Helicon Focus, where we would stack the images into one image, and then we would retouch any spots where the program failed to combine the images correctly. If we go to the next slide, we can see an example of what that looks like. It's kind of hard to see. But on the left, that is what the part of the image is supposed to look like. And on the right, you can see it's kind of see-through and blurry because the program failed to combine the images correctly. So in that case, we would have to go in with the brush tool and just kind of paint on the correct part of the image. And this is where we would also see if there was movement of the plant while we were taking photos. On the right, you can see there's some blurriness of the leaf and it has like a double edge on the outer outline of the plant. That is movement. In cases like these, our next move depended. If it was only 1 or 2 isolated leaves that showed movement, I could go in and automatically retouch them myself. But if it was multiple overlapping leaves or even the whole plant that had movement, I would have to go in and retake all of the photos, unfortunately. And then the next thing we had to start looking out for were these random sensor dots. We aren't sure why these were happening because these usually only occur in older cameras that have been exposed to lots of lights and stuff. But this was a brand new camera we were working with, so we assume it's probably because of some dirt or dust that's trapped in the sensor. So in order to prevent this in the future, we'll probably have to send out the camera to get professionally cleaned so we don't have to worry about looking for it in the retouching process. And then once we finish in Helicon, we'll export it as a TIFF file, which is a type of raw file. And we will take it into Lightroom Classic, which is where we will edit the lighting of the background and the lighting and texture of the plant itself. We utilize the masking tool for this, which is that red stuff you see on the background. It allows us to isolate parts of the image. So any edits we make to that mask only apply to the parts of the image that it's covering. So in this case, we put a mask on the background so we could darken its appearance and make it true black so we can bring focus into the plant itself. But this process is pretty tedious because the mask isn't perfect. It misses some spots between the leaves and the stems. So we'd have to go in and manually do that ourselves. And if we want it to look realistic, like it's not edited, we have to be very precise with what we're doing. So this was the most time-consuming part of the editing process. But once we did that, this is a before and after of an edited image. As you can see, I darkened the background so it's true black and the focus is completely on the plant. I also darkened the pot so it's pretty much not there. I brightened the plant, I raised the exposure, and I raised the shadows on the underside of the leaves so you could see more of that underleaf venation. So once we finished up in Lightroom after that long process, we could ship it over to Photoshop. And we mainly use this to remove any imperfections on the plants or any dust or debris that we missed on the background. So in this case, you can see I used the generative fill feature to remove those crispy brown spots that were on the leaves. We would use that feature to remove such things. And then we would also use it to remove tripod legs for the aerial images because sometimes the plant was so big we had to be zoomed out and getting the tripod legs in there was just something we couldn't avoid. But obviously we don't want that in the main image, so we had to go in and remove them. But one part of the editing we did not get to is color correcting. As you can see, these two photos have completely different tones. The one on the left is very warm, the one on the right is very cool. Neither of these are accurate representations of what the plant looks like in person, and that's something that we don't want. So if we go to the next slide, we can see we have been taking reference photos with these Kodak color control patches that in the future we can use as a reference to edit the colors of the image to make them accurately represent real life. And so they look similar to each other. So of course we encountered some challenges. Some had solutions, some didn't. These are some that did have solutions. The main one that I've mentioned already was movement of the plant or camera causing the final image to be blurry. We resolved this by creating a setup of shower curtains surrounding our camera setup to prevent any drafts from moving the plant. And then we also use timers on our camera. So in between the time that we pressed the shutter and the shutter actually releasing, there was time for the camera to settle from the movement of us pressing the shutter. We also encountered leaking water from the pots ruining our paper background, which we don't want because it warps the background. It doesn't make it smooth and even like we want to. So we handmade some little slip mats out of disposable tablecloth and black and white paper so they blend in with the background that we're using. And then for the black background specifically, we encountered a glare that we would get on the paper that made it uneven, an uneven color that is. And so we would angle the lights more to the side of the plant instead of in front. And we turned off the overhead lights to prevent any noise coming from those. And some challenges that didn't quite have quick and easy solutions involve the plant just not being ready to be photographed. You know, plants are living organisms. They're gonna have their bad days. So in this case, it was mainly just a matter of it being really droopy and needing to be watered and it not being ready to photograph. So if you look in that photo, I believe that is circulumbata. Every time I went to grab it to photograph, it was droopy for one reason or another. So I never actually got around to photographing this plant, unfortunately. But it's something that we have to work around when it comes to living organisms. And other obstacles include Glenn having to spray pesticides, which wasn't a big deal since we usually use those days to work in the office, uploading and editing images, and then also waiting for needed programs to be acquired or for materials to be bought in the early stages of the project. So things to develop in the future. The main one is microphotography, which y'all have gotten a glimpse on those posters behind y'all. These are some setups that we did early in the project and later in the project, just as we were experimenting with the equipment. If we go to the next slide, these are the lenses that we use to get such close-up images. We use a Nikon 105mm lens. That's about a 2x zoom. And then if we wanted to get even closer, we could add on that high definition converter lens on top of that. That allowed us to believe I get a 4x zoom compared to the human eye. So some other things that we want to expand on in the future are using microscopy. This will be tied into more of what you see on the poster, of course. This will include taking images of the seeds, which are really small, and maybe even reproductive structures of the flowers. We also want to delve into using the scanning electron microscope, which the garden it does have one. So we want to take advantage of using that to maybe photograph some pollen grains. And on the topic of pollen, they wish to create a pollen bank for begonias for slides to refer to back in the future. And then I recommended having multiple people walk or work on the project at once, just because photography and editing has proven to be very time consuming. And so if they wish to have the project move faster in the future, it's essential to have different people working on different parts of the project at once. So this is some other glimpses of images that Victor Mosquera took. Again, he's not able to be here today, but this is what he was working on these last few weeks of the project using his micro lens. This is what we wish to work on in the future, what we wish to present, this amount of detail of the flowers. And if we go to the next slide, we can get even more closer onto the details, the reproductive structures of the plants. On the left is the reproductive structure, and on the right is we have cut open the ovary to show the eggs inside. And something cool that I want to show y'all is this little video that we can make from Helicon Focus. It is a 3D animation where the program automatically makes a 3D image of it from the images that we stacked together. I just thought that was cool to show y'all. It could be used to put on the database for more, to allow the viewers to get a 3D view of the plants. And then these next couple of slides are just larger images of the posters that are over there by the wall, so you can maybe see the color brighter in greater detail. And again, these were both made by Victor Mosquera.
Where would you? Anywhere.
You can see some images in the top that he took using the microscopy of the seed. You can get really great detail of it and the eggs. And these seeds are very tiny. I looked at them. They're probably smaller than a grain of sand. And so to get this amount of detail was pretty interesting to look at. Yeah, that is all I have for y'all. Thank you for listening to my presentation. I hope y'all enjoyed it. I know I did.
Does anyone have any questions?
I actually have two questions. So Anastasia, one was the stacking, the focus stacking. So was that a manual effort? So did you manually move to different focal lengths?
Whenever we were taking the photos, yes. For most of the project, I would manually change the focus on the camera. But towards the end of the project, mainly when we were working on the micro photography, we started using a USB to control the camera from a computer, which prevented some of the shakiness and would automatically take the photos as needed and change the foci distances.
Okay, thank you. That's helpful. Also, a second question is, so 34 of the nearly 600 species, how did you select those 34?
I just went down the line. So I started with, in the greenhouse, all of the plants are separated based on their horticultural classification. So I just started with the rhizomatous ones, and I started at A and just worked my way down. So I believe I got to the Fs, and that's what I completed. And then I would just skip the ones that I couldn't take photos of and come back to later and such.
Anybody else have a question?
First, I just want to say that these pictures are phenomenal. You've done an amazing job, and just like working out all the kinks and like the movement and everything. That's just really impressive, and it'll be so helpful for us.
Thank you.
And then I just wanted to ask, were the problems with movement, was it more difficult with the larger begonias?
Yes, this
Okay.
one in particular, the acetosa veil, I had to retake the photos of it, I think 2 or 3 times because the leaves are just so big and heavy that even with the measures that we took to prevent movement, there would sometimes be movement. Even in the final images that I took of it, there was movement, but I was able to go in and edit it myself.
Right,
Yeah.
very cool. Thank you.
Anybody else? All right, let's give Anastasia a round of applause.
Hello, everyone. Thank you all for being here with us this morning. We appreciate everyone who's come and taken time out of their day to watch, and we're excited to tell you about all the fun stuff that we did over the summer. So today, Megan and I will be presenting on our project of inventorying and vouchering the woody plant species here at the Botanic Gardens. So first, we'd like to begin by introducing ourselves. Okay, there we go. So hello, everybody. My name is Julian, and I am one of the Living Collections interns here at the Gardens this summer. Currently, I'm working on getting my bachelor's degree in ecology at the University of North Texas. I'm particularly interested in the fields of entomology and invertebrate zoology, which I get to study under Dr. Eleanor Lichtenberg, who is an assistant professor at UNT. In Dr. Lichtenberg's lab, we study how different methods of livestock grazing affect populations of native pollinators, primarily native bee populations. And in my free time, I've also been volunteering with the Pro Museum's collections team at their warehouse in Dallas, where I'm currently working on organizing their insect collection. So after I graduate, I'm planning on pursuing a master's degree in museum science. And here I've included some photos of the fieldwork we do at UNT. We see a lot of cool bugs out on the ranches and sometimes get to say hi to the cattle. I've also included a photo of some cool specimens from the museum. On the left is a red locust, and on the right is a giant red-winged grasshopper. And you can't really see it in this photo, but that grasshopper on the right is like about as big as my hand. So it's just really cool to see in person. But yeah, I'm very grateful for this internship opportunity at the Fort Worth Botanic Gardens, and I'm excited to talk about everything that we did this summer.
All right, so my name is Megan. I'm also a Living Collections intern, and I got my associate's degree at Weatherford and then transferred to TCU to study environmental science. And I've been a plant person my whole life, and transferring at TCU, I helped to start a campus-wide tree inventory project, which I have a picture of down here with my good friends Gisela and Zoe. And then I like to think that kind of stemmed into our Southwoods project, but really it just kind of showed my professor how many people are interested in that field. So when we started working on a ecosystem services project here at the Botanic Gardens, and then me and Zoe enjoyed it so much that we started volunteering here. And then that's how I got into this internship, and it's been absolutely amazing. But now Julian's gonna talk to us about the inventory portion.
Cool.
So.
So here we've included an outline of everything that we're going to cover in this presentation, beginning with some acknowledgements, a short little introduction, and heading into the details of the project that we've been working on. And after all that, we're going to conclude by discussing some of the limitations that we faced, along with some ideas for work that we're going to be doing on these projects in the future. So first, we'd like to thank several members of the Fort Worth Botanic Gardens and the Brit team, starting with Austin Boneck, who went out in the heat with Megan and I nearly every day while we were doing this, helping us collect data while also guiding us through this internship. So Austin, Seth, Ben, Jessica, Tiana, and Zoe, we would not have been able to complete this project without you. So thank you all very much. We appreciate you providing us with your expertise. Moving on, we will briefly cover the goals of our project. So this project was actually completed in 2 different phases. The first was to fill in the gaps of the tree inventory in the north and the south woods. This part was actually initiated by a previous intern, so shout out to Savannah Thomas for getting us rolling with that. So this was just a continuation of that project and just finishing what she started by going out and creating new coordinates and also updating old ones. We also wanted to create an inventory of all the ash trees on campus for treatment purposes, which we will get into in just a little bit. And for the second half of this project, we wanted to voucher the cultivated woody species on campus and add to the herbarium's vast specimen collection. So all of this wouldn't have been possible without the use of Fort Worth Botanic Gardens online databases. We used both IRIS-BG and ArcGIS in order to locate trees and access and update missing or inaccurate accession information. So now we're going to dive into the specifics of the tree inventory project. We started this whole process at the beginning of June, and it took us about a little over a month to complete. So we'll begin with the methods we use to collect all of our data. We have tablets to access ArcGIS and an application called Field Maps, which we use to find out specifically what areas we needed to focus on with new accessions and navigate the gardens with. We also used a 2-meter GPS stick in order to tag the locations of each tree that we accessioned. And as you can see, it is a pretty large piece of equipment, but it is very useful in getting accurate GPS points, also helping us get coordinates on trees that were a little out of reach. We were in some pretty densely wooded areas at some points, so being able to reach a little further was very helpful. I've also included some photos of field maps on the tablet. So in the top left, we have a picture of a map of the entire garden where each dot represents a plant that has been, on the bottom right, each dot represents a plant that has been accessioned into our database. And you can see in the photo on the right that if you click on one of those points, the app brings up a list of all the data that has been collected for that accession. So we have the scientific name, the full accession number, what type of item it is, the status of that item, so if it's been inventoried or removed, who created and last edited that accession, the date it was last edited, and the condition of the item. So if it were a healthy tree, we would mark it as good, fair, or excellent. And if the tree was like obviously dead or diseased, it would get marked as poor or dead. And in addition to this software, and more specific to the ash tree inventory, we use measuring tape to measure the trunk diameter of the ash trees that we thought would make good treatment candidates. And this is just so we could determine how much pesticide to administer, so about 5 milliliters per inch. So once we have all this information entered into field maps, it gets transferred over to Iris, where it becomes a little more easily accessible through a desktop, and so that we can create reports from this data. And here is just a zoomed in photo of the points on that map. And moving on into a little mini outline of the inventory project, the specimens that we focused on accessioning were any adult trees that are larger than 6 inches in diameter, including both healthy, diseased, and even dead trees out in both the north and south woods, which we'll show in detail in a moment. We also wanted to focus on identifying each tree to species, grabbing GPS coordinates for each tree, and then counting up the total number of trees that we would end up accessioning. So on a typical day that we would be out inventorying, we would begin by getting all of our equipment set up, turning on the GPS and syncing it to the tablet. And from there, we would locate an area on field maps that looked particularly empty, walk over, and then start locating trees. Once we found a tree, we would identify it, input the accession number and item condition, then record any tags if they were present. And after all that data gets added to the accession, we would then record the GPS point by standing the GPS pole straight up and as close to the center of the tree as possible. And at times, canopy or cloud cover would interfere with this reading, so we'd have to move the pole around a little bit in order to get an accurate point. But a lot of the times we did get lucky with the GPS if it weren't in like a super dense area. Moving on, we've included a few maps to help better visualize what we were doing out in the field. On the left and highlighted in green, we've boxed in the area of the gardens that we were out doing accessions in. And as you can see, it seems like quite a large area, but with Savannah's help, a good chunk of that had already been covered. The map with the blue dots shows all the new accessions we added to the database, not including items that we updated those coordinates on. And as you can see, there are still a few gaps on this map, but this is mostly just due to an overpopulation of invasive species like Chinese privet or a parasol tree. which kind of took over areas that native trees would normally be growing in. So instead of taking data on these plants, we just wanted to skip them because we didn't really see a point in collecting measurements if we're just planning to destroy them anyways. So yeah, we would just start on the edge of the woods and slowly make our way inwards, kind of just navigating through the brush, getting everything that we possibly could. On the right is our updated map of the North and South Woods, which includes all of our new accessions, plus everything that had been previously accessioned by Savannah. So now it all looks nice and pretty, and we have a great visualization of all the trees in those woods, plus a database that we now get to work with. Now that we were able to get the physical mapping over with, we were able to create some charts based on the data that we collected. So on the left, we have a pie chart detailing the percentages of the top 70% of tree species that we accessioned out in the woods, with our #1 most commonly encountered species being the sugar hackberry, which we found 92 of. Additionally, we found a lot of green ash, cedar elm, American elm, pecan, and cherry laurel. Some of the least common species that we encountered were the Texas red oak, black hickory, big tooth maple, rusty blackhaw, and eastern redbud. In total, we recorded 561 accessions spanning across 30 different species. Moving on, just a little bit about the digital inventory and why it's so important for the Botanic Gardens. We mentioned this briefly before, but the collection management software that's used at the gardens is called IRIS-BG, and it has a whole range of functions that it allows its users to mess with. And we've included a couple screenshots here. So over to the right, we have a screenshot of what it looks like when you look up an item in the database by its accession number. And you can do this in a number of ways, like in the main search bar, you can type in species, family name, or its locality, and the software will give you a list of everything in the database that fits those parameters. And once you click on a specific accession from the list that's been generated, you can see all of the information that was originally entered in field maps on the tablet, and it's listed in a way that's easily viewed, and all of these fields can be updated after the fact in case, say, you missed collecting some information out in the field. You would then be able to add it in later. So right off the bat on this window, we have an accession info for a cloud forest sugar maple, and you can easily see important information such as its location, the coordinates, status, condition, the date it was collected, and who collected it. Another neat function of this software is easily creating maps of these accessions. So if you look next to the coordinates, you can see a small globe to the right. And if you click on it, Iris instantly creates a map of the area that the item is accessioned in. So with the item location marked by a little red crosshair. So yeah, this function makes things a lot easier when managing the collections. Lastly, we have a screenshot of what it looks like when we want to create a report from all this data, with each of these checkboxes marking a different filter that you could apply to the database. So this is an example of what we would check if we wanted to create a spreadsheet of all the hardy trees on campus, listing out all of their accession information. You can either click view or export, and the program will automatically open a nice looking Excel spreadsheet with all the information you need. So all of these features are incredibly useful for the horticulturalists on campus to easily track the health and condition of these trees on campus. You can create lists of items that are alive, dead, and then diseased, which could then possibly use to make inferences on what kind of species grow better in certain conditions, which is great for planning and managing the layout of the gardens. This information was also used to quickly and easily find out which ash trees we needed to mark for the emerald ash borer treatment, as we were able to create a list of trees by health and then mark all the ones in the best condition that would make good candidates for saving. So with Iris, we were able to track changes in the forest over time. So for example, it would be possible to create yearly reports of the species composition in the North and South Woods, so we could potentially find out what species are doing well, what aren't, and then possibly implementing all this data in research. So I've mentioned our ash trees and the emerald ash borer earlier, and where that comes in this discussion of our species of interest, the green ash. So a part of the inventory project was to collect information on all of the ash trees on campus, specifically the green ash, just because this is what most of them are, with maybe a couple being like white or Mexican ashes, but most of the ash trees on campus are green ash. Because if you didn't already know, the green ash is on the IUCN Red List of critically endangered species as a result of the emerald ash borer being introduced into the United States. And what the EAB does is once the adults lay eggs in the bark and begin their infestation, both adults and larvae begin majorly compromising the tree's vascular system. These beetles chew holes in the tree's phloem, disrupting the transfer of nutrients and ultimately leading to the tree's death, unfortunately. These infestations are so aggressive that ash trees all across the US are under threat of extinction. In the bottom right, we've attached a photo of what an infested ash tree looks like, and you can see like the obvious ridges carved out in the tree's exposed bark. And Bartlett was kind enough to give us 25 free treatments. So what we aim to do with these trees is choose the 25 healthiest ones that were also in locations in proximity to where guests might be able to see them so that we'd be treated. So we determined if an ash tree was healthy enough to be treated by seeing if it has the majority of its leaves that are in good condition, or if they're not like, obviously diseased. We also wanted to make sure that these trees are structurally sound, as that's a good indicator if the tree isn't dying or diseased and on its way out. So yeah, we inventoried all these trees, and with the help of the IRIS database, we were able to filter them by health and choose the ones that are in the best condition to be treated. So here we've created a map of all the 185 ash trees on campus, including the 27 that we've selected for treatment. In blue, we have the trees that have already been treated prior to this project, along with the newly treated trees marked in green. In red, we have the untreated trees, which were left either because of the condition of their health or due to being far away from any location where guests might be seeing them. So you can see we've left the majority of the ones in the north and south woods and just focus on the ones nearest to walkways. So like we mentioned, we used IRIS to create a report of potential candidates and mark those trees for the Bartlett tree experts to come in and inject their trunks with the solution of emamectin benzoate, which is a preventative of EAB that also treats existing infestations using 5 milliliters per inch of its DBH. So special thanks to Bartlett for coming out and doing that for us. And most everyone watching this presentation are plant people, but in case you needed to be convinced why it's important for us to track the condition of the north and south woods and also save more ash trees from infestation, is that trees contribute a lot, not only to the ecosystem and the health of the environment, but also to human well-being. Without these trees, we would lose a lot of biodiversity and beneficial organisms that contribute to the ecosystem services that green spaces like the Fort Worth Botanic Gardens provide. And also in terms of human well-being, trees also reduce erosion and filter pollution by trapping dust and other particulate matter that would otherwise make its way into the atmosphere and consequentially our lungs. And also in 2013, the American Journal of Preventative Medicine published a study on the relationship between trees and human health and found that the spread of EAB is correlated with an increased risk of respiratory and cardiovascular disease because in areas where these trees are being swiftly eradicated, they're no longer able to provide these beneficial services at the rate that they used to. So the main goal of this project was to make the data on these trees easily accessible so that we can track their conditions and subsequently make decisions on how to manage them so that they continue being a great resource for the environment. And now we'll move on into the voucher project, which
Alrighty, so on to the vouchering portion of this project. And then all the plants that we're going to be talking about today were vouchered on the Fort Worth Botanic Garden campus. So to start off, what is a voucher? It's essentially a collection of plant material with data connecting it to habitat, health, and ideally they'll have reproductive material. So all the ones we did collect have fruit or flowers, and then these can be used. Essentially, they're preserved on cardstock type archival paper for long-term storage in the herbarium, and they can be used they're also scanned with the herbarium for digital collection and off-site research, and then serve as a genetic snapshot into the plant's current state and its current habitat. And then it's also can be used, that's genetic snapshot can be used to track changes in phenology throughout different species across the US. So before we went out in the field and started collecting, we started developing these phenology guide charts essentially to help us decide which plants we were going to collect on and which ones would be fruiting or flowering in the summer. So we pretty much just started with, it's in alphabetical order, and we went down and found the ones that were going to be fruiting and flowering in the summer. So in yellow you have the flowering, green fruiting, and the purple would be ideally both. And these are just the ones that we did collect, except for a few select examples like the Lacana retusa down here. And that one was one that wasn't expected to be fruiting or flowering until late summer or early fall, that we found a mature specimen on campus that had mature fruit from this year and then also persisting fruit from last year. So we were able to collect that one, even though it wasn't especially supposedly supposed to be fruiting or flowering. And then this is the rest of the ones we did voucher. Another example up at the top, the Morris Elba, we were able to collect that one in the spring. And we were trying to collect the Morris Ruba or the red mulberry, but we were unable to, even though it was supposed to be fruiting into late summer, but the fruit dried up. It was much too hot for that one, so we were only able to get the white mulberry in the spring. And then most of the sources for this came from, we have like the USDA, the Department of Agriculture, the Missouri Botanical Garden Plant Finder, and then the Lady Bird Johnson Wildflower Center, along with a few select others that I linked. The sources for all the project are linked there, but I just put it on the slide in case anyone was curious about the specific sources for the phenology guides. And for the most part, they're just guidelines, and we don't know where most of the information came from. It wasn't like included in when they were like testing this plant or where it was observed. So if some of them were like observed in Missouri, it's not really going to correlate well to the Texas climate. So it's really just a guideline. And then some of them we just collected opportunistically where we're walking around, like that one has fruit and flowers, we're going to go get that one. Since we know that even if it isn't supposed to be at that time of the year. So after we had that, we were able to go out and collect data, and we were able to get 48 total trees that were vouchered in. We are also doing this on only woody trees and shrubs, so just woody species that were representations of their species, ideally with fruit and flowers. So based on our phenology guide chart, we were collecting plant material, and we wanted enough plant material from each specimen to be able to collect, have one collection for the herbarium, one for the Brit at teaching department, and then one to give away to other herbariums or research collections. And then we'd also collect the DBH or the diameter breast height. And then the height, which we did a height estimate based on our 2-meter high GPS stick, which it's kind of hard to see, but in that lower picture, it's up next to that tree, and you just kind of estimate and be like, okay, that's 2 GPS sticks tall. And then we also collected canopy coverage, sun exposure, percent dieback. We took pictures, had the GPS stick for GPS coordinates, and then we would record the location in the garden, the habitat, and associated taxa. So for the diameter breast height, it's essentially just a measure of the diameter of the tree at 4.5 feet tall, or 1.37 meters. And this graphic from the Forest Service kind of illustrates all the ways trees can complicate DBH. So like this split trunk, you'd want to move up or down, and then you can measure the diameter of both splits and add them together for total DBH. Or if there's a bulge where the split is, you have to drop up or down to get that average diameter of the trunk, basically. So we recorded that, and then we also recorded canopy coverage, where you just essentially stand at the edge of the canopy, and you stand north and take the tape measure south and measure north to south and then east to west. And that's essentially just to give us a picture of the area the canopy would cover. Or if you picture the sun at noon, where the shadow would fall for that tree. And then dieback, kind of similar thing. We're just looking at the canopy coverage, and this little graphic that I made, if you just picture that black spot as dead tree, you could just look at the canopy in like a half circle and say, okay, that's a little bit less than 25-ish%. So, and just to gauge the health of the tree. So we'd record that as well. And then sun exposure, I always like to picture the top of the tree, the canopy again, is like a pyramid, or a pyramid with like a plateau on top. And then again, picture the sun directly above it and it's where that plant would be receiving sun at noon, basically. So if the top of the tree is getting sun, that's a point. If all the 4 sides, then it would get 5 total. So if there's 2 trees next to each other, it's one of those sides is going to be covered, it's going to get a 4. Or if there's a small tree in the understory completely shaded, no direct sunlight, it's going to get a 0. And all of these parameters were recorded just to give us as much information on the voucher as possible. So anyone looking at the specimen, once it like in the herbarium or et al., it can give them an idea of the health and the environment of the plant. So you can say, okay, this one was in the understory, but it wasn't doing well, or it was doing well, and kind of use that to gauge the environment and health of the voucher that you're looking at. So, okay. So once we had all of that data, and where we were collecting our plant material in the field, we use this yellow field press up at the top, which is essentially just a bag with straps that you'd layer in your plant material. And we would layer in the cuttings that we would take in between newspaper, and you would want to get as much You want to get as many different parts of diagnostic material as you can, basically. So you'd want to get a piece of stem with leaf scars. You'd want enough plant material for both sides of the leaves, both sides of the bark if you can, and then the fruit or the flowers. While we were laying these out in the field, we would also try and like open the flowers and cut cross sections of the seeds, which is what this down here. There's like that's the jujube. It's cut, and you can see the cross sections of the fruit a little bit. But just so you can, once it's pressed, you have all those parts representing the voucher, and you can see the inside of all the fruit and the flowers. And then after it's been pressed in the field, it'll come in and we put it in the wooden press in the top right, and it'll be strapped in and compressed basically, and you layer it in between newspaper, and the newspaper goes inside foam and cardboard for like even compression. And we use the foam for bigger specimens, especially the plants with like bigger fruit and stems, so that way it doesn't, and nothing breaks, and you get even compression all the way across. And after it comes from the one press, it's going to go in the freezer or the dryer, and just looks like the middle one, filing cabinet with a fan in the bottom, where it's going to stand upright so you have airflow through all the layers of the cardboard and the newspaper, and essentially just to dry out the plant. And it's especially important if you have plants with bigger fruit, that if they have a lot of juice, you're going to want them to be really dry before you freeze them. Otherwise they can expand from like ice, but also they would mold if they were left to dry naturally. So you want them to dry out fairly quickly so you don't get mold. And then after that, you're going to move them to the freezer for a few days. And it's just to kill any bugs, pesticides, fungus, anything like that. So you don't contaminate the herbarium, basically. And if there's a lot of specimens in there from all over the world that can be susceptible to anything that you put in there. So after that processing, they come out of the freezer, and then we move on to mounting them. So we have a couple of the mounted ones. There's a China berry up top, and you can see the older fruit in yellow. This one? Yeah, over here. And then the lighter green, that was mature fruit from this year, and then you have the older fruit from last year, and then we also included a piece of bark. And if you only have one piece, you want to show the interior. So there's typically more diagnostic material on the inside of the leaf or the bark. So if you only can get one, then you want to do that. And then me and Julian are gluing them down on that archival paper, essentially a cardstock, meant for long-term storage, and then pressing them in between more cardboard and foam, and putting heavy things on them so that they can get even compression to dry, so they're secure while they dry, and for long-term storage and whatnot. So for each species, we had 2 that we mounted, and then the one that was going to saving for other institutions or other herbariums, we didn't mount that one. It's just kept as a plant material collection. And each one then also got a label with all of the data that we collected. So all the canopy coverage, dieback, all of that goes on that label. So anyone who's looking at that specimen can look at the environment and get a window into not only the genetic materials they choose to look at that, but the health of the plant and where it was growing conditions. And then on each label, we also put a QR code, and that QR code linked to the pictures that we took for each specimen. So, which were posted on INET. So we took pictures, they're posted on, and that takes us to our digital vouchers, that essentially serve as a living representation of like the scanned or the dried voucher. So up on the McClure Primifera, or the Osage orange, you can see the fruit. It's much lighter in color than it is once it's dried there on the bottom. And the seeds are much darker. The exterior and the interior have darkened when it dried, and then also it shrunk a lot. So having those pictures of each of the specimens just kind of gives a better whole picture of the voucher. So you get habitat information, and you can get living information, and then they're also available online to anyone who's not on campus, as well as the scans of the plants that go in the herbarium. They'll go in the Torch Herbaria database. So those will also be available offsite for offsite researchers. And then I also included some other pictures of the plants that we have with us over here today. So you can kind of compare colors and how things look after they're dried and processed. So we have the crabapple over there. It's got the pale fruit. It's like pale green on the inside, but after it's dried, you're really not able to see that. And then the red exterior of the seeds with the white on the inside doesn't really carry over super well to the mounted one. So you can have that preserved online. Similar with the flowers, some of them times they lose their vibrancy or they look different when they're flattened out. So we have both of those over here as well. And then I also brought a fresh Osage orange today. And then one of the ones that we froze and dried. And there's a bit of a size difference. Just like a little bit. But if you want to look at these, you can see that the like the individual pods on each of these, they're significantly shrunk and shriveled. So the pictures just give a good idea of what that looked like as a fresh specimen as well as a dried specimen. All right, so these are a map of all the ones that we did voucher throughout the season. So there's a total of 48, and mostly based on the phenology estimates that we had, each with post INAT, and they'll be in the Torch database at some point as well. And they're fairly spread out, but these are not even close to all the plants that we did visit throughout the semester. These are just these are just the ones that we were able to collect. So for the ones that, for just the trees section, we were able to voucher 41, and the 17.4% of the trees that we were able to find on campus. And then that not found category, the 17.8%, that includes plants that we went and searched for or supposedly had fruiting or flowering in the summer, but we found them and they didn't have reproductive material. So we didn't find reproductive material, or the plant had been cut down, or we just couldn't find it in general. And sometimes if there's only one of the specimen and it's a dioecious species, you're not going to find any reproductive material, or the flowers aren't fertilized. So stuff like that. We also found a few that were just like young saplings that had just been put in that were in the database. So we overall visited about 84 tree species across campus, and we're able to voucher a little less than half of those. And then the not vouchered, the 64%, those were the ones that were either expecting to be fruiting or flowering in the spring or over the winter that we weren't able to visit or get to. So for next time. And then for the shrubs, we didn't really focus too much on them for this project, but more voucher them opportunistically. And we were able to get about 7 of those. There's 3 of them we couldn't find that we did look for. And then there's about 94% of the shrub collection that's not vouchered yet. So about 4% is vouchered. And then for the shrubs, there's also a lot of cultivars across campus. So we had to be careful when you're collecting that all the plants that we were collecting, we knew exactly what they were. We knew they weren't cultivars. We knew the base species and that they were labeled properly so that we could get good information on each of those specimens that we did collect. So in total, just the combined shrubs and trees, we vouchered about 13% out of the total, and then left 86.9, so 87% vouchered, not vouchered, excuse me. And that was pretty much all the ones we could find for the summer. So we pretty much got all the ones that were fruiting or flowering in the summer months that we were here, and that were accessible to us on the gardens. So, and the goal, there's a strategic plan goal for the gardens of a 25%, which would be about 30 more plants or so, which wouldn't be too difficult in the spring. That leads us to our limitations of the project, which mainly seasonality and the time frame of that reproductive material. So we weren't able to collect a lot of them that just didn't fruit or flower in the summer. There's the occasional misidentification that we also included in that not found category, that you'd go to a plant of the same genus and it would just be like, just the species would be misidentified though. So maybe it was one we already had. So there's a couple like that we ran into. And then the cultivars with the shrubs, just had to be really careful. We couldn't really collect opportunistically on all the shrubs as we weren't exactly sure what they were. And then with the mounting of the woody species, since a lot of them have really big fruit and seeds, it made it difficult, and we had to cut cross sections on a lot of them, or you could do a separate fruit collection. And then moving on to the inventory, limitations of the inventory was the old data that Julian mentioned that we had. And then the data that we had from previous intern was mostly done on a cell phone for GPS coordinates. So we had a lot of old coordinates and the error area, I don't know. The error for the phone GPS is much larger than it was for the one that we were using. So sometimes you'd have clusters of plants that are all of the same species and you wouldn't know which data point went with which tree. So it was just a bit confusing with that one. And then the heat and the weather also kept us inside or limited our outside activity when making sure nobody got like heat stroke. And then the dense forestry, like Julie mentioned, the privet that was taking over a lot of the forests when we were inventorying. So we were able to get all of the trees that were out there, but Austin was over there with the cutters, like chopping us a path the whole time. And then the older data with no coordinates that we just don't have coordinates for, but we only have an area of the garden. So there's a lot of future work in maintaining the database, both for the sake of finding vouchers. We did use the database a lot for searching up the taxa that we had data on and going to those coordinates and finding them on Iris and then being able to check on where those plants and everything are. So there's a lot of maintaining the database that needs to be done. And then we could create or continue to use the target lists for different times of the year and hire the interns by different seasons so that they can collect each section of the reproductive material. And then that 25% goal would be about 30 more plants that need collecting. And then there's always updating old inventory and making those phone coordinates a little bit more accurate, as well as verifying identification, specifically with some of the ash trees. We're running into a lot of them. We're saying they were white ash trees, but they were just actually green. So we did some of that as well. And then finally, we included some pictures of our favorite moments, like the Atlantis February moth, yes. And then the paper mulberry, which is probably my favorite voucher that we did. Those are the flowers, and they kind of like explode outward and open fairly quickly. And then whatever that is up in the top corner, we found in the woods. It was yeah. And then also the QR code for all of our sources is linked there as well. And that includes the one for the phenology. Phenology data, which is added here as well for the rest of the project as well. And then we also have vouchers for everyone to look at if you want to see our Osage orange seeds. And then if anyone has any questions, Julian's gonna come back up here.
Any questions?
So Iris BG is already in use by the gardens, but for any of the other programs and like apps and things that you used, were those all already set for you, or did you look at some alternatives and choose?
I think they were mostly already set up by the gardens and being used before we got here. And then there's like, they can transport the data that we record on field maps from ArcGIS, and I'm not exactly sure how that process works, but all the field map data that we enter and all the data in GIS can be transferred over to Iris. So it was pretty much a setup process when we got there.
I have two questions, but I'll ask one and then give other folks a chance. Okay, could you tell me a little bit about plant identification? You mentioned that y'all found some mis-IDs. So how did you know it was mis-ID'd and how did you rectify the problem?
Basically a lot of research. There were some that I just knew from the inventory project that I'd done previously and also the inventory project on campus. The trees that I was really familiar with, it wasn't too bad. And you can recognize ash trees by the shape of the leaf scar. So it's like once you knew the shape of that leaf scar, you can just go and break the leaf off and be like, that's not a white ash. But for a lot of the ones that I just didn't know, we just were identifying them later. So just taking pictures. We were using iNat, and then also I have another plant identification I can use. And then there's a couple books that I also have that we're using. So the ones that I didn't know right off, it was just research. And then,
Okay, my second question
Okay.
is, well, I've looked around. Nobody else had their hand up. Okay, so y'all are recording different sorts of data and they're in three very different databases. You've got IRIS-BG, you've got Torch, which is this herbarium specimen data, and you've got INAT, which is the in situ images.
Yeah.
So tell me, if I'm trying to understand this tree and I want to know all the bits and pieces, how are you connecting those data? Or what do you think about that?
they're all on the labels. I definitely didn't say that, but there's a scan barcode that's going to go on the physical specimen in the herbarium, that they can, I assume, scan somehow. And then the QR code's also on that physical label. So if you're in the herbarium, you can have all of those links. I'm not sure exactly how we would do that with the online version, or if we could link it in Torch somehow. But then the INAT observations are also, it's specified which collection it is in the description of the INAT. So it's like, this is herbarium collection number like this. So if someone's looking at the INAT, they can potentially go and seek out the other ones, but it might be a little harder to find if you don't have the physical copy. So
Julian, for IRIS-BG, is there, for IRIS-BG, Julian, are these connectors provided the INAT link and the Torch link in IRIS-BG?
I know that there is a field in the IRIS-BG application for like adding the iNaturalist link. We do need to like go in and update that like after the fact, because that's not something that would like automatically be added to the database, because we need to upload them like after we come back out from the field. So that's definitely something that we should do in the future, like for future hires in different seasons, to also like add the iNaturalist links to those databases.
Hi, great work. Okay, so the vouchered percentage was, I guess, like 13 of the Woody collections. That's about right, versus unvouchered. It seems small, but it represents a bunch of hours and a bunch of work by
Yeah.
you all. So you've got a feel for kind of how slow it goes, but you also probably picked up speed as you went along and got a little better. So if we had another wave of interns doing the same thing, what do you think they could do kind of at the beginning to gain a little bit of efficiency?
Right at the beginning, like just in like unifying the process basically, or
Just from just going out and collecting, like, is there anything you could do at home or practice that would make you faster in the field, or faster? It may be that you can't go any faster than that.
in the field, I think we had it down pretty good. It just it took a couple specimens to practice, and since we're doing like 3 for each tree, it was pretty quick. The most time consuming part was probably doing the phenology charts. That original list of species that we had between the shrubs and all the trees was like several hundred plants long. So that was probably the most time consuming part. So if they already have a good chunk of that done, then that could speed it up quite a bit. But
it's also like really time consuming to like travel from one end to the gardens to the other, because you know we'd finish our voucher on one plant, and then the next plant on the list is like half a mile away. So probably just recommending that anyone who is gonna be inventorying at this position in the future should just be like physically fit and like ready to work out in the heat.
Good advice. Good
yeah.
advice.
And then the other thing is, if we travel like that half a mile to that one plant, and then it wouldn't have reproductive material, so you'd have to move on to the next one, and it was a process for sure.
Other questions?
Oh, I was gonna.
Actually, this is a question for all three of you. In terms of the skills that y'all perhaps knew about but really perfected this summer, what skills would you say you think will help you the most into the future? And it can be anything from using a GPS unit to whatever.
I think one of the skills that I would probably need to work on the most, like me personally, I don't have that much expertise in like plant identification. So that was a little bit of a learning curve for me, like getting to know all the trees that are around campus. So yeah, I think just like plant identification was one of the biggest learning curves for that.
Definitely the same for me. Plant identification. I learned a little more about how begonias are identified and classified. And I also touched up some of my photography skills, and I used a different camera for once. I'm a Canon girly, but I use like Nikon this time. So I also learned my way around some new equipment that I'll for sure use in the future.
I don't have a question. I just have a comment that another skill, you guys are fantastic speakers. And I think that the work that you've done is wonderful and that you present it very well.
Any other questions? All right, thank you guys so much. Oh, we got time.
I'm wondering if you guys have any additional ideas or thoughts about applications for the data that you have acquired that maybe you didn't get to explore in this particular instance?
I feel like for like iris and field maps, we kind of like already explored the range of functions that were like necessary for this project. I don't know if there were any other things that like I don't really know IRSPG that well, so I don't really know if there's like any other function of that application that could apply to the inventorying or vouchering project, but it's definitely worth looking into for sure.
Sorry, I just wanted to clarify, not like improvements on what you already did, but rather like looking at next steps, like using the data that you have acquired here in, say, climate research or something else. What are some, do you have any like further ideas for that?
I mean, there's a lot of like stuff that, especially the voucher information that could be used for, like we collected canopy coverage and that could be used for a campus-wide, like that's what we did at TCU was a how much of our campus is shaded type thing. So the grounds crew was using that for when and where to plant trees and which trees did the best. So it was like a similar project there. And then also a lot of it is also just for other researchers to use. So we have as much information available as possible. And if you did want to look at climate data, you have there's also fragment packets on all of the specimens with extra like material, so they could take that, microscope it, I don't know, look at the genetic data, and then they could compare it to plants across the US or plants same species somewhere else. So you could use that for identification or also tracking climate. I know there's a way to track climate like that. I don't know, but yeah.
Okay, if anybody didn't get a chance, I know that Anastasia and Victor put a lot of effort into the photography portion. I know that you all did too with the vouchering, but please go over and take a look at the posters. Those are photos of actual begonias from the collection itself, not things that were stock or taken off the internet. So when everyone in the room gets a chance, please go take a look at those.
All right, thank you all so much. The three of y'all did a fantastic job this summer. So let's give them a round of applause.